In this video, we will continue our discussion about the effect of fatty acid oxidation and fatty acid synthesis on the differentiation of human embryonic stem cells to definitive endotherms. In the previous video, we have understand that in the fatty acid oxidation pathways, the gene that is CPT2 gene plays an important role to accelerate these fatty acid oxidation pathways. At the same time, it will promote the differentiation of human embryonic stem cells to the definitive endotherms. And so in this video we will continue our discussion in the same pathway, and apart from the CPT2 gene, they find out a chemical compound or a compound that can help in the acceleration of this fatty acid oxidation or not fatty acid oxidation, and finally it will help in the differentiation of the human embryonic stem cells to the definitive endotherms. How they find out this chemical compound is when they did the PCA analysis of the cells that are treated with the inhibitors, FAO inhibitors, that is C75, and they can see that the PCA analysis lies in line with the human embryonic stem cells. PCA analysis of C75 inhibitor cells lies in line with the human embryonic stem cells gene expressions are on the same side of PC analysis. Thereafter, they analyzed the top 500 genes, and from that they did the gene ontology analysis of the pathways that are related to the top 500 genes in the PCA1 plot, and they are able to find out that it is the acetyl-CoA that plays an important role towards this differentiation or towards this fatty acid beta oxidation pathway. So what they did is they check the intracellular acetyl-CoA amount in the embryonic stem cells and in the cells that are treated with different inhibitors, and they can find out that in the embryonic stem cells the amount of this acetyl-CoA is low, whereas in the definitive endotherms, when they are treated with different kind of inhibitors such as the C75 ETO, they can see that acetyl-CoA amount is high. And thereafter what they did is they check this effect of this. They increase the acetyl-CoA, intracellular acetyl-CoA, through this chemical compound that is acetate in the cell culture medium, and with a dose dependent increase in the acetate, they can see the increase in the amount of acetyl-CoA content. And thereafter, when they check the relative RNA expressions data of the gene markers that are found, gene markers of this definitive endotherms, they can see the increase in the RNA expression of those markers such as FOXA2, SOX17, CXCR4, when both of these acetyl-CoA and these chemical inhibitors are present. And similar things they are able to confirm it using the flow cytometry analysis, where they can see that when the chemical inhibitors are used and there is no acetyl-CoA accelerations has been artificially been done, then they can see the decrease in the markers of this definitive endotherms such as SOX17 and CXCR4. However, when this acetyl-CoA is been introduced, they can see the increase in the markers for SOX17 and CXCR4. So this panel D and panel C results confirm that this acetyl-CoA plays an important role towards differentiation of this differentiation of the human embryonic stem cells to the definitive endotherms, as it can be able to rescue the effect of the chemical inhibitors. In this case, the ETO inhibitors are being rescued by this artificially increase in the acetyl-CoA concentrations, and this has been validated by both the panel C and panel D, and the quantitative analysis of this flow cytometry is also been shown in this right hand side of the panel D, where they can see the increase in the markers of this SOX 17 plus and CXCR4 with when both the chemical inhibitors and this acetyl-CoA are introduced. And similar thing they have checked when in the CPT2 knockout cells, CPT2 knockout human embryonic stem cells, and in this case also they can see that there is an increase in the acetyl-CoA content. There is in this case also they have increased the acetyl-CoA content artificially, that can be seen in the panel E. And when they check the markers for these definitive endoderms, they can see the increase in the markers whenever they increase the acetyl-CoA content in those knockout cells. For example, this is the control where there is no differentiation occurs, and this is the wild type which does not have any CPT2 knockout. And in this case, the CPT2 knockout cells has low expression of this markers, that is SOX17 and CXCR4. However, CPT2 knockout with acetyl-CoA artificial increase of the artificial increase or de novo increase of this acetyl-CoA will cause increase in the SOX17 and CXCR4 markers, which indicates that there is a differentiation occurs in the human embryonic stem cells. And these things are been also quantified in this figure on the right side of the panel F, where they can see that increase in the markers of this these definitive endoderms whenever the acetyl-CoA and CPT2 are acetyl-CoA are been incorporated, or de novo increase in the acetyl-CoA in the CPT2 knockout genes. So it can rescue the effect of this CPT2 knockout gene. So this acetyl-CoA is the main component that helps in the differentiation of the human embryonic stem cells, or it is the main component that plays a role towards this fatty acid oxidation pathway. And these things are here are being showed using immunostainings, where the markers for this pluripotent stem cells are reduced where this acetyl-CoA is used, whereas the markers for the definitive endoderms are increased where this acetyl-CoA is de novo increase in the acetyl-CoA is been done. And in the control case, we can see that there is a still high level of pluripotent stem cells markers and low level of this definitive endoderms markers. And after validating that it's the acetyl-CoA that plays a role in the that plays an important role in the fatty beta oxidations, then they decided to check whether it can compensate the role of this activin A, which is a commercially available compound that helps in the differentiation of the human embryonic stem cells towards pancreatic committed cell lines. So in this case, what they did is they reduce the amount of activin and they use the acetate to increase the amount of acetyl-CoA in the cells. And in this case, they can check the acetyl-CoA increasement with a low dose of this activin A will leads to the RNA expression of the gene markers that belongs to this differentiation is almost similar to the control case. When the C75 inhibitors are used with this activin, C75 inhibitors are used, and with this low amount of this activin A, the actual amount that is used is 100 nanogram per ml, but in this case only 10 nanogram ml is used. And this acetyl-CoA, when used, they can see that similar levels of RNA expressions for this FOXA2, and same case they have seen in the case of this SOX17, and compared to the normal case, it is significantly higher in both the cases. And for the case of this NKX1 and PTF1A, they can see higher. They can here also they can see the increase in the amount of this acetyl-CoA when the acetyl-CoA case, and compared to this non transfected case. And here this when the C75 inhibitors are used, there is a high increment with this activin 10 nanogram per molar, both in the case of NKX61 and PTFA1. And these things they have validated using the flow cytometry also, where when they use this only activin A at this lower concentration at 10 nanogram per ml, they can see around 46.1% of the markers increase. However, when they use this activin with this inhibitors such as C75 or with this acetyl-CoA, they can see the increase in the amount of this SOX17 and CXCR4. And in the quantifications also it shows the similar results, and that is activin A with the C75 or acetyl-CoA could help in the differentiation of these human embryonic stem cells towards definitive endoderms, or in this case they differentiate the cells towards pancreatic committed cell lines. So this experimental indicates that the acetyl-CoA is the important factor, and if they increase the FAO's role is to convert fatty acid to acetyl-CoA, whereas FAS role is to convert acetyl-CoA to fatty acid. So if one can reduce this FAS synthesis, fatty acid synthesis in the cells, and increase fatty acid oxidations, then they can increase the amount of acetyl-CoA within the cells, and that will help in the differentiation of the stem cells towards definitive endoderms. So this is the gist about these experimental results. And if you need to understand in detail about how these experiments are conducted and how they have analyzed their data, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.